We're traveling around the Caucasus region and selecting different varieties of ayran, cheese, and cottage cheese. Later, we extract microbes. And now we are making a dairy product for our cosmonauts at the International Space Station. So we already have some microbes. We want to improve this product. Yeah, if you could tell us, that would be great. Hello. Hello, we are microbiologists. I'm Andre. Nice to meet you. My name is Kamal. We collect various local fermented milk products. You make ayran. Could you tell me a little bit about how you do it? And if you have some, we would like to buy a small quantity. We only need a little. We boil milk. So, we cool it until it reaches 104 degrees. If we don't have ayran, we throw in some copper and close it, and it oxidizes, and the ayran ferments. What kind of copper do you use? Well, in the past, there used to be copper coins, yeah? Yeah? So we use those. But now, what do you use instead? Well, now we always have starters, ayran. All right, wait, now it is closed. Copper, that's interesting. Come in. Oh, may I? Come in, and touch it. Oh, it's warm. And how long does it take until it's ready? The next morning. The next morning it is ready. Here's the prepared ayran. Wow, how dense. Sometimes when we are making a soup without tomatoes, when we have potatoes, pasta, and so on, we can enhance the flavor. Cool. Thank you very much, Kamal. Good luck. And are these your lambs? Yes. How do they stay by this? How do they stay on the cliff? I don't understand how they don't fall over up there. Go up. Now he'll run faster. You have so many of them. Now there are 1,000. 1,000? Wow. No, really. Personally, I am hearing this for the first time. This is magic. When you don't have ayran, you add copper. What does that mean? That is, they add copper to ferment the milk. And in the past, they used coins, which were not really copper. It was an alloy. They were brass. I'm very curious what this all means. I'm interested in the potential of copper. It is quite possible that when they add copper, they are somehow influencing the potential and intensifying the process of fermentation. Or maybe the coin was just a source of bacteria, and the role of the coin itself is just magic. We are starting to move away from the medical paradigm that all microbes are evil, to the ecological paradigm that there are many good microbes all around us. Taking care of this micro-life, preserving it, we will be able to eradicate many diseases against which we currently use antibiotics. The clot is dense, formed. It is lightly sour but fresh, slightly dosed with gas.
Do you have a fridge?
No, we'll transport it quickly. Then we'll board the plane and take it to the laboratory. All right, Stas, don't drink the milk. This is a sample. How can we improve our relations with our microbes so that they protect us from bad germs, so that we don't need to use antibiotics as often, so that everything works correctly? We have come to the conclusion, after all our studies, that, in fact, we must return to natural sources of nutrition and of everything that we had. Ah, you have changed the tank. Colleagues, I want to introduce you. Once, this was Zaporozhets 969M. It has been transformed into this magical miracle. Air cooling. Look, this is a gasoline pump. We pump it with our hands, if necessary. There's an eccentric, here's a stock. To be honest, I played with such pieces of iron in my childhood. And now it helps in my job as a microbiologist. A lot of different devices. If something isn't working, I'll figure it out.
Maybe you'll drive.
I'm afraid.
There's nothing to be afraid of.
Оп. Так. Да-да.
Wow! This is my childhood. Zaporozhets. It's crazy. I don't understand at all. The formation of our civilization occurred thanks to the significant, simply colossal contribution of bacteria. There were, of course, pathogenic bacteria that destroyed entire cultures. But if we talk about such things at a planetary level, then it was bacteria that formed all that we call the biosphere. All these forests, fields, animals, because they were the first to appear on Earth, because they live in such conditions that cannot sustain any other form of life. How do we get there? This sounds somewhat sentimental, but this road, which is right before our eyes, is like science. You think it's right here, a straight, beautiful road, and see clearly where you're supposed to go. And then bang, and there's milk, and you don't see anything at all, and you don't understand anything. If I tell you in an everyday way, like, for example, how a cell divides, how it first doubles its DNA, how it initiates all the stress mechanisms, like stability, it's just the most complicated, insane system. And this was discovered very recently, something like 70 years ago. That is, there's been a colossal breakthrough, and the average person gets the impression that it's cool. Scientists have done everything, and we understand it all. But for those who work on this, it doesn't matter whether you study microbes or whether you are sending an all-terrain vehicle to Mars. You understand that you don't understand anything. Absolutely nothing is clear. And this is, of course, a wonderful gift from nature, from God. Well, it depends on your view of the creation of the universe. In my view, we are presented with a game that, for every particular individual, has no end. Absolutely no end. And the more we understand it, the more difficult and complicated it becomes. We make a lot of progress. We understand. This is the door. We open it. We open it. And behind this door, we see a tunnel that is 100 times longer than what we saw a moment ago. Having passed through it, we open another door, and we see that the next tunnel is 1,000,000 times longer. That's the nature of idea. It's the coolest thing. You just...
So this is without bacteria, see? That is, there are also beads, we shake them, but then the oil remains on the walls. And here it is being washed away. And I need something that will wash the oil off the walls completely.
I never fill out a paper without making some kind of mistake.
I don't know what's here. Yes, I have microbes that I found accidentally. They're so small that you have the sense that you've made them up. I hope they didn't die.
So, here's our task. We find some microbes that can somehow be useful. Our task is to preserve these microbes. And microbes, of course, don't like it very much when we try to dry them and preserve them for a long time. For example, if we are talking about lactic acid bacteria, they are accustomed to living in milk, and they are not used to being dried. They survive, but only a very small portion survives. And if we are talking about some kind of microbial agents, for example, probiotics, starters for fermented milk products, or the agents that we use to reduce oil pollution, we need to have a lot of microbes. Our work now is connected with this. Dania's work, and in general, our group's work, because we have found a lot of interesting microbes, and we are faced with the fact that they survive, but they don't survive very well. I would like each of you to think about what processes can destroy our cells, other than mechanical, osmotic, and oxidizing ones. What we should add, first, before drying, and second, after drying. Naturally, we need a test subject that would die during freeze-drying.
Well, in fact, Vladek K1, it turned out to be okay. K33 and K34 got stuck.
What is 34?
What's its name, gosh?
We turned off the cell. It's over. We dried it. It's in anabiosis. It's sleeping. Therefore, we only have to take care of it ourselves. As a man who works and does it well himself, he eats and eases himself. And then, when he is under intensive care at the hospital, we are forced somehow to do it for him. That's about the same thing we are now trying to develop. Some kind of IVs or whatever we have to use for this microbial cell, to keep it alive while it's turned off. The 8th and 9th. That's the whole point, that it didn't change. It didn't change, that's the point. So, then we do the following. Write it down. Let's take all 3 cultures, dry them in 2 protective environments. We give each 10 doses of penicillin. And check every 3 days. This path is beautiful, of course. When you see the finished product, and when you see that it's not just on the shelf or in the store, it's not just bringing you some profit, but when you see the reaction of someone who tells you, listen, cool, I really need this. Then he calls and says, we need the starter. Our child ate some, and now everything is fine. And when you have gone all the way from the laboratory to this kind of response, you will give anything to experience that again and again.
Моя
You have to do things that interest you. Sometimes work is just a job. You work, you do it well, in accordance with your conscience. But the motive for you is simply, I don't know, earning money and so on. But when you do something you are crazy about, then, of course, you will do it very well. And you will succeed because you like your work. Yes, that's it. Of course, at such temperatures, it is very difficult for microbes to eat oil because the oil becomes sticky. This is typical pollution: ice, oil, and water. A three-phase structure. And so we found a microbe that divides, reproduces, and eats hydrocarbon at 45 degrees Fahrenheit. That is, it can function in ice. And we have proposed a new, probably unique approach to the application of these microorganisms. We transport a lot of things on the Northern Sea route, and the cargo traffic grows every year. In 2016, a 25-year record for cargo flow was set once again. It is constantly growing. And now imagine if there were an oil spill in those icy waters. Everything would freeze in the ice. What could we do? Biological purification provides an answer to this problem. We would not need to clean up the oil spill. We could leave it to the bacteria. Let them collect it. Let them find the oil spill and let them consume it. In fact, emergency spills account for 1/1000% of what is lost along the entire route from the oil producer to the final consumer. And everything else, 90-odd%, is chronic. That's such a trifle. A little spilled here, a little dropped there. That's exactly what we offer, simply speaking. It's like a fire extinguisher that stays in place. That's how I see it. You spilled some oil. You just pour out these granules. That is precisely the idea of the agent. And it is environmentally safe. We extract the microbes here in the Arctic, and here they are returned. I had a great friend, a great American zoologist. Once when he was talking about biostations, he said that they are necessary because the work at biostations shapes a biological worldview. That's the truth. This biological worldview is formed because biostations allow you to observe a lot, and they make it possible for a zoologist and a comparative anatomist to have a conversation with a microbiologist, which is something they can't do anywhere else. That's perfect. It actually changes the mindset. This is the essence of science. Isn't that right?
Какого цвета море, какого цвета небо, какого цвета скалы, какого цвета крабы. Джа где-то здесь, Джа где-то рядом, он в этом море, в этом ветре, знаешь, Джа где-то здесь.
Sulfur. Oh, Neris. See? Neris. He's my friend. New friend. I think we should a bucket.
Comrades, do you happen to have another bucket? Just for a minute.
Где-то рядом с нами жаркое солнце, позитивная вибрация, белое море, родная биостанция. Джа где-то здесь, Джа где-то рядом.
Well, yes, unfortunately, we simply got caught up in the small, simple chain of daily events. Yeah, and in the simplicity of the algorithm that surrounds us. It simplifies your understanding in general. That is, you use a scale line. It just breaks down. It's not there. Understanding this scale, even accepting it, not understanding, it seems to me that that is not at all realistic. It is simply impossible. But accepting, it is very important in order to understand everything. I don't know. Philosophy in general, as all this is arranged, a kind of logic of living. In order to think properly with your brain, you need to clear your head. To do this, you need to sit on a block. And most importantly, you need to understand that you are sitting on a block. When you understand that completely, when you accept it, I think you will go mad.
I have started combining a heterotrophic dark consortium with subcutaneous bacteria. Zhenya is engaged in enzyme immobilization and combining this immobilized enzymes with a dark heterotrophic consortium.
What we have is a system for converting solar energy to hydrogen, but it is purely biological, and hydrogen can be converted into electricity. We are receiving energy from the sun. First, they were under such conditions that they could exhaust hydrogen, but they lived, roughly speaking, to the point of exhaustion. Now, I have given them such conditions that they are doing better. They will not exhale hydrogen, but they will be able to even out their internal balance, and they may even grow a little.
What's this? Some mold. Is it on silicone glue or what?
Cyanobacteria.
This red stuff, is it rust or something that grew here?
Something has grown here, a lot of it. So, where's that? Wow. This means that 33, and this is 34.
The story of K33 and 34 is as follows. They were cultured after incubation, although the titer was not very high. But here's everything as it is. And after drying, the titer for 34 decreased strongly. The story with 33 is this. I repeated this a couple of times.
2.
Well, to be precise, yes, it turns out that we have no colonies on the soil source at all, but on dextran and dextrose. 2 times in a row, we got an unreadable number of colonies on the source, about 300 or so, and at -1, that dropped to 0.
And you've done that again with the same result. How do we check whether the cells are alive or dead? We have a small bottle. It has a dry powder inside. In this powder, there is 1 billion cells or so. We can estimate how many cells are alive by planting them on a petri dish. If we take our powder and drop it into this tiny little cup and smear, then it should grow into 1 billion cells. And they do grow. We can't count them. We did do that somehow in order to understand exactly how many microbes there are. We used to do dilutions. That is, we take our bottle with the dry substance. Here, it is dried up. And after the dry powder has already become liquid, we take some of the substance out and we transfer it into an Eppendorf test tube. Here we had 900 microliters. We added 100 microliters. As a result, we diluted it by a factor of 10. There are a lot of microbes here too. We repeated this procedure. From here, we select and transfer here. And if there were 100 million here, then now we have 10 million, and so on. We make a series of such 10-fold dilutions from the test tube in which we already have, say, 10,000 or so cells. We can take a small drop, we distribute it onto a petri dish, and then we can count them. It's simple. In microbiology, this procedure is called a 10-fold dilution. We do them all the time. And here we faced an incomprehensible problem. From that dry powder, in which there are many microbes, there, when we plate them on a petri dish, we see continuous growth. But after we make the very first dilution, in vitro, there are 0 cells in the test tube. That's impossible. This seems like a mistake. But when the mistake is made twice, this is no longer a mistake. It is a kind of evidence that must be dealt with. Once I could not find a suitable glass for alcohol, so I took a petri dish. Well, I thought this is convenient. You can also cauterize the handle of the spatula. So, of course, my alcohol caught fire in the petri dish. Everything got very hot, and I realized that I had to put it out somehow. All I had nearby was some foil. And I had to cover the petri dish with something. Because the foil was uneven, there were some holes and the alcohol was still burning under the foil. So I grabbed it all in these gloves used for cutting roses. And I ran to the bathroom with a huge fireball in my hands. Well, I burned Andrei's chair. I didn't tell him about it until 6 months later. Margarita, where they checked for hydrophilia and hydrophobia. 33, 34. I don't understand what's wrong with them. Have you seen Danya's results with the dilutions? The system of dilutions isn't working. Before drying, everything is fine. The dilution is very clean. But after drying, the source runs off from -1 to 0. I don't know what it is.
Well, I cultured 33, so there was a lot in the source. At 1, there were hundreds, and at 2, there were several colonies.
What did you use for the dilution? Saline. Maybe sodium chlorine kills them. Some reaction starts during the drying cycle. And maybe... That's a good thought. What's happening. This may all be due to the fact that the source we transfer without dilution. And a -1 dilution is a type of dilution, like all the subsequent ones, in which our sample is in contact with saline. To make a long story short, let's do it this way. Take 33 from the refrigerator, do the dilution, not in saline, but with 30 GL sodium chlorine. But with 30 GL sodium chlorine added to the saline. And we'll see what happens. There are 2 streptococci for iron. For cottage cheese, we have to clean lactococci. I can't sleep. I toss and turn. I wonder how Andrei is doing.
I support this means of working.
Think about it for a couple of days and decide whether you are interested in going on such an adventure or not. Sleep well. You'll need that.
Okay, thank you.
And this is the first one, right? Matt method. There are no conclusions from the Ministry of Health indicating that this method is suitable, that we can do something with this method, and accept your clinical studies with this method. The most important thing is not the organization. The people who could do it are the most important factor. Zavgorodny said that the component collapsed for everyone. Apparently, they had some technical problems. Do you know what the problem is, partly? You cover the dishes too thinly. There is a mistake somewhere hidden, and we simply don't see it now.
This is really a dish.
This is a dish? Yes. Am I the only one who encountered this error? I just want to ask everyone again. Maybe I did not understand something.
There's nothing growing on the source.
Bring it here. Danya does not see his experiment. Having carried out the work, he doesn't see this as a system. Great, set it out.
Is there a chance that this is something, I don't know what?
Absolutely. We do it this way, and it all becomes good, right? This shows that some things are still unknown in microbiology. It always happens like this, always. As if some kind of power is dragging you along. So you just waste time in vain on all kinds of things. And this is the last bottle we had from the refrigerator, right?
No, there's another one.
Oh, is there another one? Danya, so what do we do with this one? It's the last one. We'll have to do some kind of ritual dance around these petri dishes. We will restore the source with Ringer's solution, and we will make the dilution with saline.
Okay.
How are ideas born? My ideas, everything that's happening in the lab. They appear from nothing. Yes, you think you can keep it in your head for a long time on this topic. But at some point, everything that turned out well turns out to have been purely spontaneous. I don't even really think, and bang! You have an insight, so to speak. How does this happen? How can you teach it? There's no way. I'm just given some materials, and that's it. I just convey them. Well, that's my opinion. And your magic core, it's here, right? Core? Your magic scanner, depth, salinity. Oh, this one. He does not want to hand it over. You still have to say, huh? What? What does it do? That is, you just shoot it down, it goes to the bottom, and on its way, it traces the pressure profile so you know the depth. I can show a picture of how this works. You downloaded it from Google, right? If it happens that someone has such a thing at the station, Pantolon had one at the Faculty of Geography. They haven't given us one for the last 2 years. Yeah, that's it. There is some tradition with such devices, I think. If they have it, they don't share it. Because that small box cost 400,000 rubles, we saw, or 200,000. Now it's maybe 400,000. Can you imagine how cool it is? Andrei Ilyich said that he is ready to go out with us on a ship, that he has one of these things. Yeah, this weather is really characteristic of the White Sea.
Did you take your sunscreen?
I didn't. For the herring, it is delicious. For us, it is interesting. At least we haven't had a storm yet. When there's a strong storm, it is impossible to work. Yeah, you need to lower the nets. The rain is pouring. You are shaking all over the deck. People are holding on like this and undergo selection.
Natural selection.
Natural selection. Who manages to stay on board? Everything should be fair. There are two types of people working here. Those who are interested and those who just do their job. For passionate people, everything is straightforward. They are absolutely crazy. They will dive. They will go to a volcano to scoop up some bacteria from the lava, and nothing will stop them. They will take your scuba diving gear. Even if you don't let them, they'll go out on their own. What we do is a kind of naturalism. We observe how animals live. This is a field of biology that has been a bit overlooked in recent years. And the observation equipment has now reached such a level that past scientists would have sold a kidney, a lung, and their liver for one shot like this. You can really achieve a lot in observational biology with such equipment. You can shoot time lapses of buried animals. You can shoot in very high resolution and observe details on something even if it is less than 1 centimeter in size. You can film animals that you couldn't really get into a laboratory, either due to the size or their fragility or something else. You can observe the behavior of animals in their natural habitat. Listen, but when you were diving, did you see these oil spills?
No, not at all.
I wonder how they actually look. It would be interesting to understand their structure. I have seen many spills, and that's precisely how this idea arose. We even tried to make a model. Paraffin balls, bang, they stick to each other. Everything is wonderful. I just don't see the bottom at all. And I want to understand how it looks architecturally, what this thing really is. Is there any microbial growth on the surface, or anything else?
Listen, if I'd shot this, I think that this photo would have been bought by the largest companies 28 times already. So, we have large invertebrate organisms. They can subsist on chemical substances, but they live in symbiosis with chemotrophic bacteria, which have colonized their insides from head to toe. And basically, these bacteria consume chemicals, and these bacteria are used either as food or in symbiosis, the bacteria secrete something that can feed these invertebrates directly.
I think, you know, the next level of agents for the removal of marine oil pollution is not bacteria, but invertebrates. You can fill them with some consortium, find suitable microbes, place them there, and voila!
That would be cool.
You know, it's like an armored infantry vehicle. There are troops inside, and it's moving so fast, because the bacteria themselves are being washed away, while the invertebrates are fixed in place. It's great. Listen, we have to think about this. By each drop.
These are devices in which electrogenic bacteria live. That is, simply speaking, bacteria that produce electric current. This system works like a battery. As long as we add fuel, it will produce current. And we can provide fuel constantly, half-eaten food, wastewater. If such a system is made very effective and very large, it can power a light bulb.
Bacteria can influence the stimuli in the form of an electric current differently. Here we have a positive charge, but here there will be a negative charge. But we can change them. We will turn a negative charge into a positive charge and vice versa. We will recharge it and see what happens. The problem is that there are no mathematical models for microbial fuel elements, and it's very difficult to calculate. And so, for now, all we do is observe, yeah?
Yes, absolutely. I suggest we start...
Well, the experiment cannot fail. We just don't know whether the expectations are justified or not. There will simply be 10,000 experiments fewer to achieve the goal. Well, everything is very strange anyway, of course. In fact, you can conduct a simple experiment. We no longer have these cells, of course, but you can make a microscopy. Well, he added Ringer solution without salt, diluted it, and plated the cells. That's exactly what we should do, because they are usually inflated mercilessly in the usual Ringer. We will even see it visually. They are unlikely to be visible, in fact. Such a gift is rare, to see the whole mechanism clearly, beautifully and visually. I doubt that they are really torn apart, that they are just dying. That happens very rarely, although it would be nice, of course. To humiliate. Then we will finish with all off 33. If it tricks us again somehow, we will just burn it all and forget it. If it starts screwing with us, we'll just burn it.
I got it.
Look, you will do everything, everything, all the plating. You have a bottle with the source and 3 Eppendorfs. You take 100 microliters of one, 100 microliters of the other, and 100 microliters of the third. You do that and then immediately go to make the agent. And then you immediately look under the microscope. We have a few minutes to see this, to see the dynamics of what is happening. 1784, living with bacteria. Let's have a look. We're looking at the culture of the microorganism. Or rather, unexpectedly, the microorganisms, which have been freeze-dried. The fact that we saw small colonies on the dishes, large colonies, all that shows is that the culture is dirty. Curiously, 2 microbes died right away. That is amazing, because in theory, one was supposed to die, and the other was supposed to survive. And it is quite possible that these thin, long ones are alive, or vice versa. The fat one is alive, and the thin one is the one that was torn apart, already torn apart. At the same time, I see such thickening at the ends, as if they were spores or something like that. Heads. Yes, they really look like heads indeed. These are not heads. These are cytoplasm. These are torn cells. Yes, these are torn cells. Here it is clearly visible. Here it is, in the center. This is something that got out, and this got out, and this got out. Are we really this lucky? Now you will see beautiful torn dead cells. The bulging eyes of microbes. Interesting.
And this.
They are being torn apart, and the balls come out of them.
You think so?
I'm sure. I saw it. Well, they are clearly torn apart. This is beautiful. Look at these balls. Look at this with your own eyes. It is a lot better here. So once again, here the dry powder is plated after the drying stage. Next, we make a series of 10-fold dilutions so that we can count the cells. What should we see? Here, in this cup, there should be quite intensive growth. Here and here, there should be nothing. And there, there should be growth at a rate of 10 times less than here. But here it is 10 times less than here. That is the logic of this experiment.